Good morning, everyone, and welcome. I'm delighted to have you here today. Today's presentation is titled Nanotechnology for Enhanced Drug Delivery and Bioavailability. We'll be exploring how nanotechnology is transforming drug development, delivery, and product performance. Over the next few minutes, I'll walk you through the latest advancements and practical insights in this exciting field. So, let's get started and see how this innovative approach can make a real difference in healthcare. Today, I want to talk about how nanoscale science, specifically in the 1 to 100 nanometer range, is transforming pharmaceuticals. When we apply nanotechnology, we see significant improvements in key areas such as solubility, stability, permeability, and bioavailability of drugs. These enhancements mean medications can be more effective and work better within the body. This technology also opens the door to more precise medicine, allowing us to target specific cells or tissues, which reduces side effects and improves outcomes. It enables targeted delivery, so drugs reach exactly where they need to go, and it can even lower the required dose, leading to fewer doses needed over time. Nanotechnology is particularly supportive of advanced therapeutic areas like biologics, peptides, vaccines, and oncology treatments. These types of therapies often face delivery challenges that nanotech can help overcome. Overall, this integration of nanotechnology into drug delivery is a major step forward in making treatments more effective, personalized, and safer, really aligning with the broader goal of improving patient care through innovation. Many APIs, or active pharmaceutical ingredients, tend to have poor aqueous solubility, especially those classified as BCS, II, and IV. This means they don't dissolve easily in water, which can really limit their effectiveness. Traditional dosage forms often struggle to overcome these solubility issues, leading to inconsistent absorption and therapeutic outcomes. That's why there's a growing need for targeted, controlled, or sustained release systems. These approaches can improve how the drug is delivered, ensuring it reaches its site of action more efficiently and maintaining effective levels over time. At the same time, patient compliance and overall therapeutic effectiveness are crucial. If a medication is easier to take and works better, patients are more likely to follow their treatment plans properly. However, when dealing with complex molecules, conventional technologies often fall short. We need enabling technologies that can handle these sophisticated compounds and improve their bioavailability. And this is where nanotechnology comes into play. It offers innovative solutions to these challenges by enhancing solubility, enabling targeted delivery, and ultimately improving patient outcomes. That's why nanotechnology is so essential in modern drug development. Let's start with lipid nanoparticles, or LNPs. These are crucial carriers for delivering mRNA, cRNA, and other nucleic acids. They protect these sensitive molecules and facilitate their entry into cells, which is essential for gene therapy and vaccine applications. Moving on to polymeric nanoparticles, these are valued for their controlled release properties and biodegradability. They allow us to fine-tune drug delivery, releasing the therapeutic agent slowly over time, and eventually breaking down safely within the body. Next, we have solid lipid nanoparticles, or SLNs, along with NLCs, which stands for nanostructured lipid carriers. These platforms are known for their high stability, making them ideal for protecting and delivering drugs that need a stable environment. Then there are nanoemulsions and micelles. These are particularly effective in enhancing the solubility of poorly soluble drugs. By improving solubility, they enable better absorption and bioavailability, which is a common challenge in pharmaceutical development. Moving to inorganic nanoparticles, such as gold and silica. These are mainly used for imaging and therapy. Their unique physical properties allow for precise imaging and targeted treatment, making them versatile tools in nanomedicine. Finally, we have nanocrystals. These are tiny crystals that increase the dissolution rate of poorly soluble active pharmaceutical ingredients, or APIs. This enhances drug absorption and effectiveness, especially for drugs with low solubility. Together, these platforms form the key toolkit in nanotechnology for advancing drug delivery and medical imaging, helping us develop more effective and targeted therapies. Now, let's take a closer look at some of the key manufacturing technologies used in nanomaterial production. First up is high-pressure homogenization, a technique that applies intense pressure to break down particles into nanoscale sizes, ensuring uniformity and stability. Moving on, we have microfluidization and microjet milling, which utilize fluid dynamics and high-velocity jets to achieve precise particle size reduction. Next, solvent evaporation and diffusion methods are crucial for creating nanoparticles by removing the solvent around a dissolved substance, leading to controlled particle formation. Nanoprecipitation and self-assembly are also important. They rely on the spontaneous organization of molecules into nanostructures, often driven by changes in solvent conditions or concentration. We also see the rise of continuous nanomanufacturing, especially in the context of pharma, where continuous processes improve efficiency, consistency, and scalability in pharmaceutical nanomaterial production. Lastly, spray drying and lyophilization are essential for stabilizing nanomaterials. They help produce dry powders that are easier to handle and store without significant loss of their nanoscale properties. All these technologies are interconnected, each playing a vital role in advancing nanomaterial manufacturing towards more efficient, scalable, and reliable processes. Now, let's delve into some critical quality attributes that influence the effectiveness and safety of our nanocarriers. First up is particle size and size distribution. These are fundamental because they affect how the particles interact with biological systems, including cellular uptake and circulation time. Consistent size distribution ensures reproducibility and predictable behavior. Next, we have zeta potential, which is essentially the surface charge of our particles. This influences stability, as well as interactions with cell membranes and biological molecules. A well-controlled zeta potential helps prevent aggregation and prolongs shelf life. The polydispersity index, or PDI, gives us an idea of how uniform our particle sizes are. A low PDI indicates a narrow size distribution, which is desirable for consistent performance across batches. Encapsulation efficiency tells us how much of our drug is successfully loaded into the carrier. High efficiency means better utilization of our active ingredient and potentially lower doses needed. Drug loading capacity is related, but focuses on the amount of drug per unit of the carrier. Optimizing this ensures we deliver enough therapeutic payload while maintaining stability. Surface functionalization is about modifying the particle surface to target specific tissues or cells, or to improve biocompatibility. This customization can significantly enhance therapeutic outcomes. The release profile and stability are crucial because they determine how the drug is released over time and how stable the formulation remains under storage and in vivo conditions. Controlled release can improve efficacy and reduce side effects. Finally, all these attributes feed into what we call critical quality attributes, or CQAs. These are the key parameters that define the quality and performance of our nanocarrier system, ensuring they meet safety, efficacy, and regulatory standards. By carefully monitoring and optimizing these factors, we ensure our nanocarrier platform is both effective and reliable, aligning with the overall goal of delivering targeted, safe, and efficient therapies. Let's talk about the key factors that influence our process, which are known as critical process parameters or CPPs. First up is mixing speed and intensity. This is crucial because how quickly and vigorously we mix can affect the uniformity and size of our particles. Next, we have homogenization pressure. Applying the right pressure ensures consistent particle formation and prevents aggregation. Temperature control is also vital. Maintaining the correct temperature helps us manage solubility and stability throughout the process. Then there's the solvent to antisolvent ratio. Getting this balance right impacts precipitation and particle size distribution. The concentration of polymers or lipids in the solution is another important factor, as it influences the viscosity and ultimately the characteristics of our final product. pH and ionic strength are also critical because they affect the stability and solubility of our components. Process time and shear forces shouldn't be overlooked either. These parameters influence the duration of interactions and can affect particle formation and stability. Overall, understanding and controlling these CPPs is essential because they directly impact the quality, reproducibility, and efficiency of our process, tying everything back to our overall goal of producing a reliable and high-quality product. Let's delve into how we identify critical quality attributes, or CQAs, as well as critical process parameters, CPPs. This step is essential because it helps us understand what factors truly impact the quality of our nano-manufactured products. Next, we utilize design of experiments, or DOE, to optimize our formulation. DOE allows us to systematically explore various formulation variables and find the best combination for consistent performance. It's a powerful tool for refining processes efficiently. A robust control strategy is also key to maintaining consistency across production runs. By establishing solid controls, we can ensure our product quality remains stable, even in the face of minor variability. In addition, we incorporate process analytical technology, or PAT tools, for inline monitoring of nanoscale attributes. This real-time monitoring enables us to detect issues early and make adjustments on the fly, which is especially critical at the nanoscale where small changes can have big effects. Finally, all of these elements fit into the broader framework of integrated lifecycle management under ICH guidelines Q8 through Q13. This approach ensures that quality is built into every stage, from development through manufacturing and beyond. And connecting this to the overall theme, applying quality by design in nanomanufacturing helps us systematically understand and control our processes, ultimately leading to safer, more effective products. Let's explore the regulatory expectations that are shaping the development of innovative nanomedicines. First, the FDA guidance and EMA reflection papers emphasize the importance of detailed characterization and thorough safety assessments. This means we need to understand exactly what our nanomaterials are, how they behave in the body, and ensure they are safe for patients. Biodistribution and immunogenicity considerations are also key. We have to carefully study where these particles go in the body and how the immune system might respond to avoid unintended effects or immune reactions. Now, these regulatory insights are particularly relevant for a wide range of applications. For example, mRNA vaccines and gene delivery systems often use lipid nanoparticles, LNPs, which require precise characterization. In oncology, targeted chemotherapy benefits from nanocarriers that can deliver drugs directly to cancer cells, minimizing side effects. We're also seeing promising developments in oral nanoformulations for poorly soluble APIs, which can improve bioavailability and patient compliance. Other applications include transdermal, intranasal, and ocular delivery systems, expanding treatment options for various conditions. And let's not forget regenerative medicine and diagnostics, where nanotechnology can enhance tissue engineering and disease detection. Overall, understanding and meeting these regulatory expectations is crucial for advancing safe and effective nanomedicines across these diverse applications. Looking ahead, the future of nanomedicine is incredibly exciting. We can expect AI-enabled nanomedicine to revolutionize how we design and optimize treatments, making them more precise and effective. Automated continuous nanomanufacturing will enable us to produce nanomaterials at scale, consistently and efficiently, addressing many of the current production challenges. We're also moving towards multifunctional hybrid nanocarriers, which can deliver multiple therapeutics or diagnostics simultaneously, enhancing treatment versatility. Personalized nanotherapeutics and digital twins are set to transform patient care by tailoring treatments to individual patients and using digital models to predict outcomes more accurately. However, as promising as these advancements are, we also face significant challenges. Scaling up production remains complex, and current analytical methods often struggle to keep pace with the needs for quality control. Stability and reproducibility are ongoing concerns, especially when moving from lab to clinic. Plus, regulatory frameworks vary across regions, which can slow down development and deployment. Ultimately, addressing these challenges will be crucial to fully realize the potential of these innovative approaches and bring the benefits of nanomedicine to patients worldwide. For further support, please don't hesitate to contact us. We're here to help you with any questions or additional information you might need. Thank you so much for your attention and engagement throughout this presentation. Remember, we're committed to assisting you every step of the way, so feel free to reach out whenever you need.